Thank you for coming to our talk. Drew DeMarco and I work on HealSync together, and Drew is the lead engineer, and I'm the lead product manager. We work for the Utah Data Coordinating Center, a full-service academic research organization at the University of Utah. We developed this tool for production use and are excited to share it with you. James has lived with sickle cell disease his entire life. He knows the cycle well. Pain that comes on suddenly, emergency room visits, and medications that help but never fully resolve the discomfort. Some people describe the pain as sharp, stabbing, or like being electrocuted, and others compare it to broken bones or a rubber band snapping repeatedly. SCD is a group of inherited blood disorders that affect hemoglobin, the protein in red blood cells that carries oxygen. In SCD, red blood cells become abnormally shaped, crescent-shaped, and rigid, leading to various health complications. Managing SCD pain is complex, and while opioids provide relief, they come with risks. Like many others with SCD, James hopes for better, safer treatments that address not just the pain, but his overall well-being. In another hospital, a newborn named Mia is being carefully monitored for neonatal abstinence syndrome. Newborns exposed to opioids in the womb are at risk for this condition. Symptoms can include tremors, excessive crying, irritability, and problems with sleeping, feeding, and breathing. Her symptoms are manageable, but her care team faces a challenge. There is no universal standard for the best treatment approach. Each hospital may handle cases like Mia slightly differently, making it difficult to build a consistent body of knowledge on what works best. James and Mia represent two different aspects of pain management, yet both highlight a broader challenge in clinical research. How do we ensure that studies are producing data that can truly inform better treatments? Think of a research study like a recipe. It follows a set of steps to collect data, just as a recipe outlines how to cook a dish. But if every study measures things differently, like using different pain scales or tracking symptoms in inconsistent ways, comparing results becomes nearly impossible. A protocol is the structured plan for a study, defining what data is collected and how. A data element is a specific piece of information, like a patient's pain level or medication use. Without standardization, studies operate in silos, making it harder to build on past research. The NIH HEAL initiative is creating a common research language through common data elements, or CDEs, so data can be seamlessly compared, combined, and applied, driving faster, more reliable solutions for pain management and opioid use. Drew and I recently spoke with Dr. Kathy Sword, an investigator and bioinformaticist involved in multiple HEAL clinical trials at the Utah DCC. As we design and develop HealSync, it's essential that we deeply understand the problem we're addressing. We needed firsthand insight into how researchers currently extract data elements from protocols, a time-intensive process that diverts focus from their primary research. Dr. Sword provided a detailed walkthrough of her workflow, and today I'll demonstrate this approach using two synthetically generated NIH HEAL initiative protocols, Momentum for sickle cell pain management and Cradle for neonatal abstinence syndrome. Here's an example of a researcher's workflow. In the upper right corner, we're reviewing Momentum and scanning for elements. There isn't a single protocol template, each one varies. In Momentum, elements are listed under study procedures, while in Cradle, they're referred to as assessments and appear in section 9 instead of Momentum section 7. Despite these differences, the researchers' goals remain the same, identifying key elements from both protocols. On the left, we see a manually created spreadsheet crosswalk. The first column lists the required HEAL domains, while the second and third columns contain extracted elements from each protocol. The researcher manually copies and pastes these elements into the respective columns. In some cases, additional transformation is needed. When a protocol describes an element differently from the HEAL standard, the researcher must adjust it accordingly. This is where HealSync comes in. Our AI-powered system automates the extraction and harmonization of clinical data elements from research protocols, ensuring faster, more accurate standardization with NIH guidelines. By integrating HealSync into the research workflow, we allow scientists and clinicians to spend less time on manual data tasks and more time advancing treatments. In essence, HealSync bridges the gap between research and impact, ensuring that discoveries for conditions like SCD or NAS and many more reach patients just like James and Mia faster. HealSync scans, extracts, and structures research variables, maps and validates key elements across protocols, and it produces a well-formatted crosswalk spreadsheet that includes matching elements for each protocol. And this tech stack is the foundation of HealSync, an open source system designed for structured data extraction and harmonization. While this system was built with researchers in mind, the ultimate beneficiaries are patients like James and Mia, along with the clinical trial participants who will benefit from the efficiencies we've created. Our research team validated what many, including Dr. Swart, have long experienced. The traditional manual analysis of protocols is extremely time intensive. Tasks that once required over 20 hours of expert work can now, in some cases, be completed in under 5 minutes. HealSync eliminates the tedious process of reading protocols and manually extracting data, freeing up valuable research time. From the moment a document is uploaded, the system automates the entire workflow, delivering structured, actionable outputs. Most importantly, HealSync ensures consistency and accuracy across all protocols. With that, I'll turn it over to Drew, who will walk you through exactly how this works.
Sure, let me pull that up.
Drew, let's start by uploading a protocol and asking HealSync about it. Okay, great. So it gives concise, high-level information. When assessing eligibility for a clinical trial, patients must meet predefined criteria. Let's ask, what are the inclusion criteria for the protocol? Okay, perfect. HealSync is extracting the specifics. In this case, the patient must be between 6 and 17 years, along with other key requirements. The results are thorough and precisely aligned with the protocol. Let's take it a step further, extracting data elements from two protocols and generating a downloadable crosswalk. Okay, so in seconds, we have a structured crosswalk ready for analysis. Drew, can you open that now for us? So as you've just seen, HealSync transforms hours of manual protocol review into minutes of automated precision. This isn't just about saving time. It's about accelerating the entire research pipeline from protocol to patient impact. By eliminating data harmonization bottlenecks, we're helping the HEAL initiative maximize its investment in cutting-edge research for pain management and addiction treatment. Thank you.